Now that we talked about damage to proteins, let's talk about damage to another important macromolecule in the cell, DNA. The most important molecule inside a cell that is vulnerable to damage is arguably DNA, the molecule that stores the heritable information within the nucleus. Its structure, first photographed by Rosalind Franklin and described by Watson and Crick, consists of two strands of phospholipase sugars running from a phosphate end towards a hydroxy end, connected to nitrogenous bases. These two strands run antiparallel and are held together via hydrogen bonding between these nitrogenous bases. These Watson-Crick base pairs can only form between an adenine and a thymine base, as well as a cytosine and a guanine base. Each strand, therefore, is directly complementary to each other. Just like proteins, the chemical functional groups in these molecules are subject to chemical reactions. The phosphate sugar backbone may get hydrolyzed, leading to DNA strand breaks. Amino groups may get hydrolyzed to carbonyls, and alkylation or oxidation by ROS or RNS may chemically alter nucleobase structure, which leads to base mispairings. DNA may experience damage from environmental factors as well. When UV light from the sun or a tanning booth hits DNA, it may lead to the generation of covalent bonds between two adjacent thymines in the DNA strand. Alkylating agents, such as nitrogen mustard from the chemical warfare agent mustard gas, are able to attach hydrocarbon groups to nucleobases. Oxidizing agents can create oxidized nucleobases, and even two entire DNA strands can be cross-linked with each other at guanine bases. In addition to these environmental sources of DNA damage, normal cellular processes can create damage. Of course, our old acquaintances, ROS and RNS, are in the forefront here once again, leading to oxidative modification of the DNA bases. The water in the watery cellular environment itself may hydrolyze off amino groups or break the bonds between the nucleobases and the ribose sugar. Cells even have their own alkylating agents, especially in the form of a molecule called S-adenosylmethionine, or SAM. Usually SAM, a highly reactive molecule, is tightly regulated, but may also react spontaneously and in a non-regulated fashion with DNA. Finally, the cellular enzymes that copy DNA, called DNA polymerases, are error-prone, sometimes incorporating the wrong base into the newly copied DNA molecule, or even slipping out, leading to large deletions or insertions. When DNA becomes damaged, the resulting lesion may be repaired by various cellular repair mechanisms. If the lesion cannot be repaired, a mutation is the consequence. Mutations result in insertions or deletions of bases, or the presence of a wrong base. This in turn may alter the primary and thus the tertiary structure of the affected proteins, possibly with functional consequences. But even if the lesion is not in a protein coding region, DNA damage can be extremely problematic. Any outcome of damaged DNA may include double-stranded DNA breaks, which is the worst-case scenario for DNA and the cell. Whenever double-stranded DNA breaks happen, the normal repair mechanisms are unable to properly fix the lesion and simply try to connect the broken pieces to wherever it may fit. This may lead to severe chromosomal abnormalities. The DNA double-strand break response, moreover, may include the induction of senescence or even apoptosis, when even these error-prone mechanisms of repair fail. Various DNA repair mechanisms exist that are designed to deal with each specific lesion. The principles, however, are always the same. Specialized proteins recognize the lesion, leading to the excision of the damaged piece of DNA. Using the undamaged complementary DNA strands as a template, the missing piece of DNA can now be filled in, which repairs the lesion. This principle is best illustrated in the case of base excision repair, or BER, and DNA mismatch repair, MMR. BER gets initiated when a base has been damaged by chemical modification, or altogether lost by hydrolysis from the ribose sugar. Specialized enzymes recognize the lesion and, if needed, hydrolyze off the damaged base, creating an AP site, in other words, a site that lacks a base. AP sites allow for the recruitment of endonucleases, which can cut the phosphate backbone of the damaged single-stranded DNA molecule, which in turn allows a DNA polymerase to come in and now fill in the gap. Mismatch repair, or MMR, in contrast gets activated as a consequence of replication errors due to DNA polymerase malfunction. Similar to BER, specialized enzymes recognize the base mismatch due to the faulty geometry of the non-Watson-Crick base pairs that are present now. And this, in turn, leads to the degradation of a piece of DNA on either side of the mismatch. Just like in BER, DNA polymerases then fill in the gap using the undamaged DNA as a template. These principles also apply to nucleotide excision repair, which gets activated when large structural distortions are found in DNA. These three aforementioned DNA repair mechanisms rely on an undamaged complementary piece of DNA to fill in the damaged gap. But sometimes DNA damage is so extensive that this is no longer possible. Therefore, a workaround has to be found. One such workaround is the interstrand repair or translesion repair. Especially when replication is stalled, for example when cross-links exists between two complementary DNA molecules, specialized endonucleases then remove the lesion altogether. However, since the problem involved both strands of DNA, a template strand no longer exists to fill in the gap. To repair the lesion, the damaged DNA therefore invades the corresponding region within the homologous chromosome, which then can serve as a template for the repair. Finally, even double-stranded DNA breaks can be repaired as part of the catastrophic repair mechanisms. If the DNA is damaged in a way that creates overhanging ends, these ends can then be used for homologous recombination, similar to what we just discussed in translesion repair. In this case, the overhangs invade the homologous regions on the homologous chromosomes, which then can serve as a template for the fill-in by DNA polymerases. At the end of this process, two tightly linked chromosomes will have been created, forming a so-called holiday junction, which will have to be separated. If that separation is faulty or unsuccessful, once again, chromosomal abnormalities may be the consequence. The other scenario is a clean or blunt double-stranded DNA break that contains no overhangs. This leads to the recruitment of the Q proteins, which simply initiate the joining of blunt ends, called non-homologous end joining, or NHEJ. Since there is no need for a repair template, any blunt end can be joined to any other blunt end. If there are too many blunt ends to choose from, chromosomal fusions may occur. In both cases, double-strand break DNA repair is initiated by the ATM protein, which serves as a checkpoint protein for DNA repair. As discussed in earlier modules, ATM in turn can activate p53, which then serves as the final gatekeeper of cell fate, potentially initiating senescence or apoptosis. As is the case with unrepaired or insufficiently repaired protein damage, DNA damage has significant consequences for cell and organ function and aging. Older individuals, by and large, show larger amounts of DNA damage, mutations, and chromosomal abnormalities. However, there is quite a bit of variability attached to this observation, as environmental factors, even fairly trivial ones such as the amount of exposure to sunlight, can significantly alter the amount of DNA damage accumulation. Thus, skin aging may be affected by UV exposure, lung aging may be affected by exposure to another DNA-damaging agent, cigarette smoke, but the effect by which either drives organismal aging may be less clear. Moreover, endogenous factors too play a role, such as the level of antioxidants or the activity of our repair pathways. All these factors vary from individual to individual, and even from tissue to tissue. Nonetheless, by and large, older individuals and older tissues show greater amounts of DNA damage, especially in the form of double-strand breaks or oxidized nucleobases such as the aging marker 8-oxo-7,8-dihydroguanine. Even more dramatically, mutations in genes for enzymes involved in DNA repair very frequently show accelerated aging phenotypes, as well as developmental abnormalities. Not only that, since there are inter-individual differences in the activity of DNA repair mechanisms, correlative analysis has shown an ever so slight correlation between greater repair activity and longer lifespans. Most importantly, though, when the activity of DNA repair pathways are compared in tissues from older to those of younger individuals, older tissues not only have generally lower activity, but moreover fail to respond as vigorously when challenged with DNA-damaging stimuli. Highlighting the importance of DNA repair to aging are studies that show that interventions that increase longevity often improve DNA repair mechanisms. Similarly, model animals that were engineered with mutations in DNA repair systems often also show progeric phenotypes. For example, reduction of nucleotide excision repair activity in C. elegans and mice leads to shortened lifespans, as does reducing the activity of non-homologous end joining. However, not all models of impaired DNA repair lead to accelerated aging phenotypes. Likewise, progerias, either naturally occurring or in laboratory models, do not recapitulate all aspects of aging. This suggests that while DNA damage may be an important accelerator of aging, it is clearly not its singular cause. Before we move on from the symptoms and molecular causes of aging to anti-aging interventions in the final module of this course, I'd like to briefly talk about another DNA-related aspect of aging, that of epigenetic regulation of DNA. Epigenetics is the study of genetic changes that are not due to changes in DNA sequence, but can still be inherited to subsequent generations. In other words, there's information content in DNA that is not contained within the nucleotide sequence itself. The most important epigenetic change that can occur is DNA methylation, the addition of a CH3 or methyl group onto cytosine bases in DNA. The molecule that donates this methyl group is SAM, which we talked about earlier as a natural alkylating agent. Usually, the cytosine precedes a guanine base, which is why these methylatable DNA sequences are also called CpG islands. Methylation of CpG islands is usually associated with gene silencing, that is, the inactivation of the affected genes. CpG islands are predominantly located in non-coding regions, such as gene promoter regions, telomeres, or centromeres. Interestingly, the methylation status of CpG islands is maintained after cell division, while the newly synthesized DNA daughter strand is unmethylated right after replication. The parental DNA still carries the methylation modification. Using the methylation pattern of the parental strand as a template, DNA methyltransferase enzymes then copy this methylation pattern onto the daughter strand, thus maintaining the epigenetic status of the new DNA and therefore also of the new cell. This is critically important for proper tissue identity, since methylation leads to the shutting down or silencing of large genomic regions. Cell identity is governed by which genes are active within a cell. A liver cell has a certain set of genes active, but a kidney cell has a different set, and so on. If methylation patterns were wiped out with each cell division, tissues would be unable to maintain only their own cell types within the tissues, and organs could no longer fulfill their function. Hence the need for maintenance of methylation status. Another epigenetic modification acts on the level of histones. Histones in eukaryotes are proteins critically involved in DNA organization by tightly binding DNA around themselves. Histones may be chemically modified as well. The two most important ones are acetylation and methylation. Just as with DNA methylation, histone modification is an epigenetic event that may silence gene activity. Together, these epigenetic modifications allow for tight regulation of developmental programs, but also enable an organism to respond to changing environments. For example, learning has been associated with epigenetic modifications. During a lifetime, the epigenome may undergo similar changes as the DNA itself. These so-called epimutations thus may alter wholesale activity of genes, which could conceivably contribute to aging phenotypes. Even if no epimutation occurs, a whole array of environmental stimuli can change the epigenomic landscape. Some, early in embryogenesis, may lead to severe developmental abnormalities. Other, later in life, may lead to cancer and other diseases. Interestingly, childhood trauma often is associated with epigenetic changes that are associated with PTSD or depression. During aging, DNA generally becomes hypomethylated, with hypermethylation in a few select regions, especially hypermethylation of tumor suppressor genes. This may in part explain the prevalence of tumors with age. Since the major methyl group donor is SAM, and SAM metabolism is linked to cellular redox state, this provides another link between oxidative damage and aging. In addition, other diseases of aging, such as Alzheimer's disease or atherosclerosis, have been linked to an altered epigenome. The science of epigenetics is still fairly new, and even newer is the study of epigenetics in aging. It is one of the most intense new research fields. While not much can be said with certainty, epigenomics, with its mechanisms of wholesale gene regulation, may provide yet another promising avenue for interventions in the aging process. Check your knowledge. Describe types of DNA damage. Explain the various DNA repair mechanisms. Explain the connection between DNA damage and aging. Define epigenetics and describe epigenetic modifications. Explain the role of epigenetics in development and aging.